Okay, Harold, as we continue sort of our discussion about your decades of experience developing neuromodulation with a special focus on depression, this is a part I'm very excited about to really talk about the birth of TMS, and how you describe it in the shadows and the wings of ECT. And so I think it bridges very nicely with what we said before. But before that, I actually wanted to ask you about something. It's something you mentioned in the previous parts of this interview, and you've told this to me before. And I'm sorry to say that it took me a couple, sometimes you get an idea and it has to kind of enter and it doesn't, the impact of it doesn't hit until you're like, you're about to go to bed and then you're like, so you said something very profound that I want to kind of start with going back to it, which is that you talked about depression as something that we're wired for, not as not as a pathology, but something that is actually an evolutionary advantage. So can you restate that idea and then what does that mean as far as treating depression and then we'll go and get into TMS.
Yeah, I think fundamentally from a neuroscience point of view, either you're a release phenomena when it's something this complex and integrated, or you're a deficit phenomena, or to some extent you could be both. But that's from a neuroscience point of view, and that's a fundamental for circuit thinking. And there was good reason for people to think that the depressed state reflected was like aphasia, the interference with ongoing function, because it resulted in deficits in movement and appetite and sexual activity and so on. And so what you were doing in terms of restoring function and using that type of thinking would be very different, because then it would be rehabilitation type of treatment.
So in that model, it's almost like an injury that you need to heal.
Right, exactly. And the other model, and this I think is runs through a lot of our thinking about pathology, is and it's less seen often in neurological circumstances, is a release phenomena like a seizure and automatic speech. And so the issue was, is this a affective organization, a normal state that's gone wild in some way, uncontrolled or uninhibited, or was this really a disturbance in the expression of this state? And obviously it could be both, but that was essentially my thinking and the phenomenology I think was there. And also what seemed extraordinary about mood was that the triggers could be abstract, propositional, that ideas could trigger mood, that what we thought mattered in terms of our feelings.
Yeah.
And that doesn't have to happen biologically. That they're street. That our moods could distort our thinking, and our thinking can distort our moods, and that's where psychological treatments essentially have a space. And but at the biologic level, it's always being expressed in terms of some circuitry. At least that's how I've been thinking about these phenomena. And in ECT, what was profound there was that you could set a dial on a machine, change the amount of charge you were going to deliver, and have 10% of people get well, or 70%. That was a proof in your face, so to speak, in life, that the underlying circuitry that was being altered was fundamental to the experience. It could be modulated that way. And of course, that's always been one of
When you say proof, you mean that there was a biological substrate
yeah,
to
that
the
you
disease, because obviously the stimulation wasn't talking therapy. The stimulation wasn't asking them about their mother.
right,
The stimulation
exactly.
was doing something to their brain.
That in a positive way, you could alter people's mood, and that the circuits go both ways. That thoughts can change mood, and that the biology of mood will change the thoughts too, and in this complex world we live in. But that simplicity, because you could do a real experiment and figure out circuitry, was very powerful for me in terms the sort of the understanding that this has much wider implications for the field of neuromodulation.
But even going back to what you initially said, even from a circuitry perspective,
Mm-hmm.
I guess you could imagine it as we have a depression circuitry,
Mm-hmm.
and it has now been, like you call it, released in a perhaps pathological way,
Right.
versus we have a circuit and now that circuit is broken, and that's why we shouldn't be sad. It's broken, and that's why we're sad, right? Those were you still have that
Right.
aspect of it that you're talking about.
Right. And the fact that you could inhibit circuits, that in fact, that was our language and our thinking at the time. And it was very simplistic in many ways. And it was the work I was doing was some of the first circuit-based thinking in terms of how it underlies emotion and how there's breaks and so on. But that all predated the connectivity perspective. It was essentially the same type of sort of mechanistic approach. And I think it has a real reality. I mean, is it's actually achieved some proof.
Mhm. Um, all right, but now sorry, now I want to take it another little step up before we get into TMS. Where you're talking about understanding that depression is not just that it has a biological basis, right? And so now that makes me think about this discipline or field of biological psychiatry. Can you explain the rise of that as a discipline, and specifically alongside of ECT, and then perhaps leading to the role of TMS?
Well, yes, a little bit about that. In many ways, the biological understanding of the major mental illnesses was the original academic approach. I mean, that's where we have the Kraepelins and the Alzheimer's and that real tradition. It was a neuropathological tradition. It was a tradition that the fundamental perspective of that field was that the major mental illnesses, both mood disorder and psychosis, were either degenerative or they were congenital. That there was nothing you could do
Right.
to alter the path of these illnesses. That you had no there was therapeutic nihilism. And that was universal across biological psychiatry, so to speak. And what upset
Including for mood.
including for mood, because the only treatment, for instance, we had biologically for mood, and the Freudians and Freud was sort of a later, somewhat later, and a separate path that we could talk about. But within the biological frameworks, we were treating the mood disorders with barbiturates, sedatives,
Mm.
and cold presses, and the average length of stay was 2 years for hospitalizations for mood disorders and so on. They were profoundly impairing. You can imagine bipolar disorder in particular. And it was ECT that changed the whole world in terms of the perspective that you couldn't do anything, because with the advent of ECT, tens of thousands of people who were institutionalized were discharged. Going back to the community, it was like night and day for many of these
And when
people.
is this?
This is in the
Timeline.
40s, in the
40s.
late 30s and 40s. And it ushered in the pharmacologic revolution, right?
Mm.
And it was ECT, in fact, that the antipsychotics and the antidepressants were tested against to see how did they stack up in terms of
That's interesting.
you know uh,
Okay.
right, and then interest in ECT itself proceeded academically, very little with the introduction of the drugs. It was sort of like, but it was the drugs that because of their incredible widespread use. I mean, when you think of it, that the percentage of the American public that is on a psychotropic is quite extraordinary, and the fact that it was the introduction of the drugs that ushered in a modern science of clinical trials. And this is across medicine. That was one of the impacts of the psychopharmacology, was that the need to develop reliable diagnostic and outcome assessments and the importance of randomization and many of the things we take for granted across all of medicine really was spurred on by the advent of the psychotropic medications and NIMH establishing units, assessment units, basically, to promulgate modern scientific methods. So I think that also was important in spurring this biologic revolution, as well as, from the scientific point of view, the discovery of the catecholamines and the underlying biology and so on that created a whole different framework. And American psychiatry was, sort of had this battle with a psychoanalytic tradition starting around the 20s and 30s, because that became a very powerful voice within psychiatry, of many of the major departments essentially being totally psychoanalytic in its expression. And that was to many extent true at Columbia and where I was. And just before I came, the new chairman of the department, who was Ed Sacker, who had discovered the first biological abnormality in depression, phase advancement of the release of cortisol. He did the first 24-hour urine samples and so on. He and Don Klein essentially fired all of the analysts and transformed that institution. So those types of battles went on in many, many places, and the field went over in that direction.
Okay. All right. So now that's very interesting. Okay. So let's talk about TMS. Let's talk about electrical stimulation that does not produce seizures, yet can treat a variety of disorders, including mood disorders. So, from your perspective, how did TMS emerge? What were the big milestones?
The very early history of TMS it's fascinating, and I think the highlights are pretty clear. The idea of trying to induce current in brain without pain had been around for quite a while, and so some of the very important basic work being done by Merton and other people to map out the motor system, basically, had to use painful electrical stimuli.
Right. So again, this would be electrodes placed on the head. You're
Right.
connecting it to a stimulator that can produce an amp, but unlike ECT, you keep the frequency low, so you're not producing seizures. But you
Right.
can do things like produce these involuntary motor. But I've had it. I mean, I think you've had it too. It's it's torture level painful, right? It's rough.
Right. And there's no doubt that ECT stimulators can be used for torture. That they're capable of because of the magnitude of the stimulus that they would provide. So that was an incredible.
But to be clear, if the people are asleep, so there's no pain.
Yeah, during
Modern
ECT
ECT.
itself, there's no pain at all.
Painless in that sense, yeah. Okay.
Right. And that was always a huge impediment to the study of stimulation, is that you can't get enough of it in the brain to do anything without it being painful in the awake individual. And going into the late 1890s, there were attempts to generate sufficiently powerful magnetic fields by Maxwell's equations to induce an electric field that would do the same thing. And they were successful, the decades and decades ago, in producing phosphenes, which is likely due to the fact that the retina has extremely low thresholds.
The phosphones would be flashes of light, and again, it's in a sense it's still not transcranial because it's still peripheral, right? You're just just activating the
It shows that they did something, and the weakest possible thing that you could do, and it was the electrical engineering problem, which fundamentally, in many of the things that we do, that is the thing that we come up against is a field, and that's where the progress has really been moving the field is at the technology end, is giving us more and more powers to do things that we never thought we could do. And Tony Barker's instantiation of that at Sheffield was really a brilliant illustration of the persistence and the innovation of a single individual who was not in an academic setting, who had profound respect for and interest in the history of science, and thought it was possible, and so he did it. And this is that is my understanding is that Tony was a physicist in a hospital setting, and he had an academic appointment, but this was not his primary labor. And and for what it's worth, I recently was asked to write a letter of recommendation for Tony to be knighted. Was very happy to do so.
So electrical stimulation going through the skull, too painful.
Mm.
Barker builds really the first modern example of a transcranial magnetic stimulation device, a TMS machine. This is just one pulse at a time, right? But it does the trick in that where, now, for example, you can produce involuntary motor twitches by stimulating the motor cortex, but without the pain
Right.
that is associated with the electrical approach, right? And that's this is in the UK. So that's low frequency, but you're proving that you can get through the skull now.
Yeah, and what was brilliant and to me of great interest about that approach was that you had greater control of the over the electrical field than you would with transcranial electrical stimulation, like in ECT. That going back to the ECT work, my work had strongly indicated that the modulation we were doing of prefrontal tissue was fundamental to the efficacy, and that ECT didn't work when you were triggering seizures from the motor strip, for instance. That's how low dose right unilateral my theory has always been. Is the reason it was ineffective is that it was the wrong site in seizure triggering, and that the motor cortex has the lowest threshold in the cortex for seizure activity, and so by lowering the dose there, that's where we're getting it from. And there had been work in the 40s in ECT doing bi-occipital, which resulted in visual defect, as you would expect, and so on. So that the TMS offered a tool to do the same thing, but to do it with greater precision of dose because of the cleanness of of it not being essentially distorted by the scalp and the skull.
Right.
So that was the beauty of the invention to me.
So for example, if you do the TMS over the motor cortex, produced a twitch, a small displacement of it, forward or back, and the twitch would go away, reinforcing the notion that, this technique was relatively focal. And by putting the coil over different parts of the brain, in theory, you could stimulate those different parts of the brain. And I think this notion that magnetic is focal and electrical is less so stayed with us for a long time. I think that's probably a topic for
Right.
the true limits is another interesting discussion. All right, but how do you get from, oh, look, I can do this and produce a twitch, right? Which I guess they're now doing in London, and Rothwell and Merton are picking up that technique. How do you get from that to a treatment for depression?
Well, you have somebody like Mark George intervene. I mean, to tell you
Mhm.
how you do it, because what was evident really early on was that when you did this repetitively, you could have either an excitatory or inhibitory effect on tissue. That was the first fundamental claim, and that was on the basis of imaging work largely. Um, and so that the frequency of the stimulation itself was causal in terms of either producing opposite impact on metabolic rate and blood flow, and presumably it had different neuroplastic effects. So type of work also seemed to be confirmed at the preclinical and the animal, some of the animal work that was being done.
So now the next step is, instead of applying a single pulse,
All
you
right.
apply trains of pulses, tick, 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 tick. And depending on the rate, the frequency of those pulses, which could be faster or slower, there was evidence that would suggest that you could either increase the function of the brain, whatever that means, increase excitability, or
Yeah.
depress brain function. So now you had an intervention that could sort of tune brain excitability.
Right. And it's interesting that that was a metaphor, and there was exciting data that was somewhat in support, but you don't see a continuance of that approach, and instead the metaphor is now shifted to connectivity, which, and just as an aside, to me, I've been a critic of the connectivity stuff, even though much of the imaging work I did was among the first to look at covariance patterns and ask which regions speak to which regions and so on. Largely because of the nature of the measures that are being taken are with fMRI. The bold response is so limited in its dynamic range, and there are many factors that will impact on dynamic range, that looking at the correlation between regions when you only have a plus or minus 2 percentile dynamic range seems to me very, very artificial. So it's one thing to say that blood flow has decreased in a region, and another thing to say, well, its correlation with another region seems to have changed. But,
All right, so be as it may, we now have TMS machines. We have TMS machines that they have some rate. Tick, 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 they can go to it. And we have the idea, right or wrong, that depending on how quickly you set that clicking rate, you might be able to even, increase brain excitability, whatever that means,
right, or decrease it,
or
right?
decrease.
Right, and
But that still is not
and
treating
then
depression.
you had demonstrations of two things with that. You had experimental demonstrations that you could do things like speech arrest, interfere with
Oh,
just get a twitch, essentially do functional mapping with it, and so that made it look like uh, it was capable of modulating important activity of the brain in a way that had behavioral outcome, and um, and you produce seizures. Um,
okay.
that that to me was a highly attractive adverse event, to the extent that under some conditions this technology was capable of taking normal individuals. These are often subjects in psychology experiments who had a seizure, so that it might be useful for the deliberate induction of seizures as well.
So this was an adverse event, not of single pulse TMS, but of repetitive TMS. And even to today, this remains the main concern with TMS. And a lot of the safety protocols that exist around TMS are designed to sort of as robustly as possible prevent the induction of seizures. So we can get back to doing it on purpose, right? But the general framework remains TMS,
Mm.
multiple pulses, and very intentionally do what you can to actually avoid seizures, right? So it's a sub-seizure intervention. Um,
That's the hope of it, or it could be above or sub. It's an issue of electrical engineering. What are you giving? And
okay, but still, with all these pieces in place, to decide that this could be used to treat depression requires there's a leap that has to be made there, right?
well, I think the leap that Mark made, because he was really the first to not only study it, or he was among the first, I should say, because there was a host of labs that were doing this at the very beginning. Um, Basically the field of TMS went to fast left, slow right, dorsolateral prefrontal cortex targets. And they were very fortunate because both worked.
Okay.
And both worked against sham. I mean, to this day, if you want to say where can I reliably know what the target would be for depression, and they were crude in how they got there, and so we could go through all of that. But it was a incredibly fortunate set of choices, and there were a bunch of choices that were made in this. Um,
So there, the notion is frontal left mean fast, higher frequency in order to try
right.
to enhance
Exciting.
or boost. Right, slow, meaning lower frequency, supposedly to depress. So the field moves in this direction. But who thought that that would treat mood or
Well, there had been, remember, as I said, when I started ECT work, ECT research, it was with the idea that there was a fundamental lateralization to the nature of emotion. And largely that work and the work of Davidson at the time, we're talking back, major papers are in 78, at the time had raised the hypothesis that hyperexcitability on the left was associated with euphoria and on the right with depression, and that you could inhibit either of those to treat those states. And that was, in fact, the type of thinking that brought me into studying ECT originally. So it was,
And this we talked about in the first, I think the very
right,
first part of this interview. Okay,
right,
this lateralization and
right.
okay.
Well, that I don't believe it.
Okay.
I think that was a gross oversimplification, but the interesting one. But that infused the field of TMS from its beginning, and stays to this day. And as I think I said in that previous interview, the fundamental study, the 2x2, where you credibly go slow, fast, left, right, and you randomize, has not really been done. And I think it would disprove and be of clinical relevance mechanistically, because then it would have to change totally that we think that fast or slow matters in these ways.
Okay, but still at the time, Mark George and a small group of people
And,
around him decide that there's an imbalance issue in depression, and excitatory to the left or inhibitory to the right will correct this imbalance, right? That's sort of the state.
and part of what helped with that is that there may have been, and it's amazing how we forget our history, maybe 20 or more PET SPECT studies quantifying blood flow or glucose metabolism, showing a negative correlation between how depressed you were and metabolic rate in the left prefrontal cortex, particularly dorsolateral tissue. It doesn't mean that it wasn't also on the right, because a large number of studies also showed it there, and my work showed that the bilateral decrease was associated with getting better, the bilateral prefrontal and so on. So there are a fair amount of things that are embarrassing to that perspective, and choosing that and making it frequent. But it was a good choice.
In Richley was. Okay, but so around the same time, and I think Mark George was one of the earlier people actually involved in fMRI and functional imaging. So he's also seeing what he believes are these functional imaging based evidence for some sort of lateral dysfunction.
Right. And among the things that was convincing to me is that an hour after an ECT treatment, all the changes you got in the regional cerebral blood flow, and with absolute quantification, were on the side that you simulated, were on the right side.
Oh.
There was no lateral, whereas you could produce lateralized effects galore in terms of the impact on memory and affect and so on, but it was clearly that you could do those things through functional changes on one side of the brain. Um,
So Mark George gets up one morning someday, right? And he's thinking about all these things, all this history. He's aware of it that you brought up and this imaging work. And he says, let's start doing excitatory, repetitive
he,
TMS to the left
he,
side of the head, and that's going to treat depression. Is that
Mark is a fellow with Bob Post at the NIMH, and a research fellow. He had the very, very good fortune of being at Queen Square in London when Tony Barker lent John Rothwell one of his very first devices, very first to really show effects in the human. And Mark was on an elevator at Queen Square where some guy said they just did the weirdest thing to me, make my fingers twitch by putting something on my head. So he immediately went to Rothwell's lab and observed it, and came back to the US and worked with Bob Post, and Bob was the chief of biological psychiatry at that division at NIMH, and I'm the chief of biological psychiatry at Columbia. So we're sort of in sister places, and it was in visiting Bob's lab that I met Mark the first time. And
okay? That was the first time you met.
I
Okay.
met him, and I met Bob Greenberg, who is also a fellow,
Okay.
and who has done great things in neuromodulation. So that's where we met, and Mark was committed to the idea that you could modulate circuitry in this non-invasive way, and that if you gave enough of
And without seizures.
and without seizures, yeah. And you can imagine that did not endear him to a good part of the psychiatric community that thought that ECT was not just the way, but the only way. And so that generated a lot of disbelief and
So the reception was not warm to
from
the.
some of the community. And that's sort of, I think, very common that we sort of become identified with our fields of study or our therapeutics that we specialize in, and that we unionize that it's them and nobody else, and that is very common, I think, in our field. So
But the pushback from the ECT field, let's say from the ECT establishment against TMS, was partially related to the fact that the seizures were considered essential and therefore a sub-seizure therapy could not work? Was that the crux of it, or it was just they just didn't like something new?
the idea that some type of stimulation of the brain that did not result in seizures would have therapeutic properties was either rejected as impossible, it was heretical, or it was very unlikely to succeed. Let me put it that way. A lot of skepticism associated with it, and part of it, I think, was, and you see this to some extent to this day. I sort of had a middle of the road position there to the extent that in relation to ECT, there's no doubt that when TMS came out, it was therapeutically pipsqueak. It did not have anywhere near the therapeutic, and I wrote editorial in Biological Psychiatry, saying essentially that it was not yet at prime time.
TMS was not yet prime time.
Yes, yeah, and but that wasn't the issue necessarily because it was finding its footing in terms of who was it useful to treat and how do you use it to treat and so on. And when we did the first meta-analysis of the therapeutic properties of TMS, we published that with Burt and Lisenby. Essentially, the conclusion was this is a really interesting intervention. It's clearly better than sham and has antidepressant properties, but they're incredibly weak. Um, that was sort of and I maintained that position after TMS was approved by the FDA, even though I was a senior author on the study that led to its approval. And Mark and I were the co-PIs on the OpTMS study, which was the independent industry independent study. Um, and in terms of depression, both of those studies essentially found the remarkable thing that they found is that only 5% of patients were remitted with sham intervention, even though you were coming in daily for treatment, seeing somebody daily. They had the lowest placebo, compared to any drug trial. Placebo rate was lower, yet the contact was so much higher. And the remission rate was around 15% at the primary endpoints. And 15% ECT is, around 70%, at least 50 to 70% remission rate in a much more resistant population. And so honestly, when looking at those outcomes, my impression was it may be of help to some people, certainly because it's non-invasive. We've done some of the early studies on its cognitive effects and found that there were no adverse cognitive effects. But I wasn't jumping for joy. And what has happened is that TMS, and this is a really important, I think, lesson, has matured to a point where it is as good or better, I would say, than any medication. That and it's only getting better. It's a moving target, but right now, in with data on let's say 50,000 people who've gotten TMS. I chair the registry data for Neuronetics, and they have data now on about 175,000 people who've been treated with TMS. The outcomes are, more than double what they were in those initial studies. And so we're talking about 60 to 70% responding and 35, 40% remitting. And so it has become, really a very useful treatment. And then the last thing in this is, in epilepsy, if you fail two anticonvulsants, you're a candidate for surgery. Because we know enough about the sequential treatment that the likelihood of benefiting substantially from the next anticonvulsant is there. We're getting to that point in depression where the likelihood of sustained benefit after so many failed trials is
Drug trials.
drug trials is very, very low. So this, and of course, the labeling for TMS and VNS and now esketamine were the first times the FDA ever labeled anything as a level of its treatment resistance. That
It's part of the label that you have to.
yeah, all of these treatments have essentially introduced treatment resistance as a subgroup, and how do you assess that, and so on. And we don't know where TMS stands there. I'm hoping to move it more and more as a treatment of first choice because of the lack of side effects. I think it's an accident of history that it was tested in people who have failed medications to begin with. So I think its therapeutic potential is even greater. And then the last point is TMS is remarkably lawful in a way that surprises me. That the efficacy as a function of how many treatments you've had and where in the treatment sequence you have. We're really now defining essentially the functions that
Mhm.
are associated with clinical benefit with this intervention, and it is remarkably stereotypic. And so that gives us all sorts of, to give you an example, in a sample of over 7,000 patients, we showed, now replicated in multiple other samples, that uh, when you start TMS, you're improving at a rate of about 3% per treatment.
Mhm.
Then after 5 to 10 treatments, it drops to less than 1% per treatment and stays that way to the end. This is with standard TMS where you're giving it one session a day. And so the big question is, why is there that big drop? Can you accelerate that? Can you rescue for that drop? And all sorts of things that will define the future of this treatment. How it has matured in terms of being, there are now more TMS providers in America than there are ECT providers. It's another.
One thing you've told me I think is very important, I want to underline it, is the fact that the TMS as it was born, there was constant innovation and there remains today constant innovation to make the treatment more effective. And so the reason we have the TMS we have today is only because there was this aggressive and ongoing push to do it. And I would sort of, on the one hand, I would contrast that with areas like, for example, TDCS, where I think unfortunately that push hasn't happened. And so we're still working with the original, right? And that is certainly a missed opportunity as far as optimization. But also to contrast that with ECT, we're talking about, I think in the 2 parts ago of this interview, there was a bit different. You had the potency to begin with.
Right. I was
It's the
thinking.
side effects that you were optimizing to.
Right, but ECT is a great example of where we did the reverse. That it started at high intensity, a stimulus that had to be deconstructed in order to make it safer. What happened with TMS was because of the seizure concern, it started off with the weakest approach to treatment and has slowly and somebody like Nolan Williams has accelerated the field, to use a pun, because he said, I'm going to give it everything I got and see if I get a faster and more successful treatment. So when he went from one session a day to 10, from 600 ITBS pulses to 1800 in the sessions, on and on, neuronavigation. So piling it all on into one. That wasn't done or couldn't be done. And that's the same thing with TDCS, I would think, that it hasn't pushed the limits to know what the limits are.
You have the therapeutic window, right, between no potency and then and the side effects and your type. And it's not, and the dose space, as you're saying, it's not just intensity. It's intensity, it's session duration, session number, personalized, all these other things. You're titrating that space to give you that therapeutic window. Okay, now we moved ahead. Now we know TMS is a very effective intervention. You've just documented the reasons. That wasn't clear when Mark George and company were first proposing this idea. So I just want to go back to that time point. If you could recount to me, you told me the story before about some of the pushback that Mark George got at NIH and maybe how and describe that pushback, also what had to be done to overcome that pushback.
Yeah, sometimes when you are taking what seems to be an original path, others seem to have really pretty strong ridicule. And when Mark was a fellow at the NIMH, I was there a decent amount of the time because I had the job of being executive secretary for a blue ribbon committee that was charged with essentially recommending reorganization of the NIMH. NIMH was the first of the intramural institutes to undergo this type of outside review where everything about it, its mission, its the quality of its research, its educational components, clinical components, budget certainly, are all under review by an outside. And we actually wrote a book on recommendations on what to do with this. And so I was in Washington when the brand new director of NIMH, I won't mention who, one could look it up historically, but this new director, who I had a fair amount of contact with, given the role that I had there, had an all hands meeting, his first meeting with the intramural program. And it was everybody at the intramural program had to be there, and it was in the auditorium there. And I happened to be sitting next to this fellow by the name of George. Mark
Young
George.
fellow at that time, right? Okay.
Right. Uh, New director of the institute announces pridefully that NIMH will no longer support idiotic research that is simply a waste of the taxpayers' money. And as an example of this research, we've been doing research, he says, that attempts to link mood to areas of the brain using imaging, and that's phrenology, total phrenology, and an example of a total waste of taxpayers' time. And the perpetrator of this fraud was a young fellow by the name of Dr. George. And Mark
He
was
calls them out for the imaging, not for the
and
TMS.
not for the TMS. It was the imaging, I believe, that was, and I happened, I knew Mark. We had discussed TMS at this point, and so on. So I turned to Mark and said, this is not a bad thing. Everybody now is going to read your work. They want to know what this is about, and so your name is out there. But you can imagine it would be traumatic for anyone to hear that, particularly from the head of your institute. And as I understand it, Mark was not allowed to talk about much of his work. He was instructed not to speak to the press. I mean, he was muffled, and presumably that's one of the reasons why he ended up going to MUSC, going back, is because of the reception he received in Washington.
But the first TMS treatments are being done at NIH, correct? And what
Right.
was the
And Bob
reception?
Bob was extremely receptive of Mark's work, enthusiastic about it. And Mark had the advantage that Mark Hallett, a very esteemed neurologist, and in fact, Mark became our first deputy editor for neurology at Brain Stimulation.
Yeah.
He was at the Neurologic Institute doing work with TMS.
But so as these, I guess, as the first papers or maybe even the first abstracts are coming out of TMS for depression, do you remember what's the reception among the broader psychiatric community? Was it excitement? Was it, I don't know, attack, criticism?
Um, I don't remember colleagues coming up to me and saying, "Hey, did you hear about this thing called TMS?" That there wasn't a lot of jumping up and down. I think one has to remember that the initial work that was done was often small sample work. The early days, for instance, there are a slew of studies of 10, 15 people per group given active or sham for 5 treatments or 10 treatments, claiming some degree of difference in therapeutic effect, often of small magnitude. So there was a lot of skepticism. Is this real? Is it not real? A lack of real discussion in the community as to why this might be. What the mechanistic level, how could you explain it? And the ECT part of the community to this day, sort of has not integrated it that well. So the journal of these ECT and other neuromodulation treatments has always had sort of an ambivalent attitude about the other neuromodulation treatments, how they get represented in that journal and so on, and at the meeting.
You think so? You're saying maybe the adopters of TMS are not necessarily the ECT practitioners
I
then
think a
now?
lot of now I think there's a lot of overlap. There's when I go to the meetings, and I think the whole idea of interventional psychiatry is being an umbrella that subsumes these different interventions is a wonderful reality that, and at least in terms of my original discussions with Mark and some of the stuff we wrote about, my thought was if I had believed that the reason ECT is so effective is because it's enhancing inhibition in prefrontal structures, and it's doing that through an anticonvulsant mechanism, the triggering of the seizure to stop the seizure results in release of GABA and all of that stuff. There's no reason why you couldn't do a similar thing more gently, where a pulse results in an increase in an inhibitory effect that you build up over time. That I
Yeah.
didn't I didn't ever think that there was only one way to get to that same type of end. And I think that that is sort of was key. And the other thing that, again, my view is that no matter what intervention we're talking about, I still think we have to be remarkably humble, and that we're only 3 or 4 steps beyond bloodletting. That even though we have these new effective interventions,
These work, to be clear, but these are working interventions. Okay.
working interventions, but there are a lot of people who still are being left out in the cold. And so that's why, for instance, when we started the journal, it was explicitly saying that we had to be agnostic to technologies. We can't get into these intersign warfares if my technology is better than your technology, and so on.
All right, so in the next part of this interview, I do want to talk about the journal. I do want to talk about maybe some broader issues in other technologies we haven't touched on and the future direction. I want to finish maybe with one question that maybe sets up that as far as bigger issues.
Sure.
The transformation of TMS from a academic exercise to an FDA approved treatment involved a company, right? A company had to come in and invest and sort of not just invest some money, but maybe be very intentional about the direction of the field and the type of trial you needed to run to satisfy the FDA. What are your general thoughts about the role of industry, about the role of companies, specifically in brain stimulation?
Um, I think it's very difficult for a scientist to invent a molecule.
Mm.
But it is not difficult at all for a scientist to have profound impact on neuromodulation technologies. I think you don't see a lot of, individual labs that produce a drug that ultimately benefits mankind. Um, and I think that if you're doing innovative work in the neuromodulation space, you can't not be involved with a company that 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 in one way or another. I mean, I spent 10 years designing with MagStim the first stimulator capable of deliberately producing seizures in the human, MST. And with certainly Mecta and Sigma Stem,
The ECT companies, yes.
the ECT company, I had custom devices made by them, and we did RCTs with them, many of them funded by NIH, and that immediately fed into what was available to the clinical community. And when we did we now call the O'Riordan study, the pivotal study with Neuronetics to approve TMS, that was where the medical director at TMS, Mark Demetrek, wanted to import much of the methodology we used in ECT clinical trials. It's non-trivial about how you, for a procedure, how you organize your trials and what you do in terms of maximizing reliability of measurements and how you can, evaluate durability of benefit, and it goes on and on and on. And so And so if you want to try and give that technology the best shot and the most rigorous shot, which is sort of should be the aim of companies as well, particularly if you're going to FDA, then that's largely going to come out from the collaboration with academia who have this level of expertise.
So
And
this
just
is
to
the
finish,
collaboration, is what you're saying. It's not the academics, it's not the industry, it's the collaboration that pushes things forward.
oh, absolutely, yeah. That you try and make. I mean, it's hard to be agnostic and also collaborate with particular industries, but that I think is the attitude you have to take. That you're playing it down the road, and you want calling balls and strikes as they are, and it's also the goal of the company that you're collaborating with is that they want the outcomes and the most rigorous way possible. When we did the optimization of TMS with Neuronetics. I'm sorry, with Neuronetics and the James Long Company, what we did, our contribution in that trial was to do the first active sham, to
Mm.
use electrical stimulation to produce the same pain under a TMS coil, but without the TMS coil discharging, so that nobody would know whether they're having a discharging coil or not. And so in that trial, we tried to markedly increase the rigor of sham-controlled trials in TMS using the same auditory input to the treater as to the patient, blocking any of the external sounds. With Vahi Amassian and Mark George in my basement lab, we figured out how you can send an electrical stimulus, the pain associated with an electrical stimulus to a particular spot. And so we added that to, and Neuronetics collaborated with us because we had the Mecta Corporation, the ECT company, build a stimulator that went down to 2 milliamps, what TDCS gives,
Yes.
right? We were in the 0 to 10 milliamp range with a Mecta stimulator
For the sensation part, for the
for
sensation.
the sensation part, but it was triggered by the discharge or not of going to the Neuronetics.
I see.
Right? So the two companies had to have devices that spoke to each other, right? So we could so that was where having positive relations with both companies resulted, I think, in benefits for the field.
Okay. I think it's a perfect place to finish this part, and we will resume with a bigger discussion of where the brain stimulation has gone and where it's going and the foundation of the journal.
Okay, great.